In the last objective, we gave an overview of the signal transduction that occurs in the main types of receptors. And so in this objective, we focus on the G protein coupled receptor and the events in signal transduction following second messengers. All right, so the second messengers we're going to focus on are cyclic AMP, IP3, diacylglycerol or DAG, and calcium ions. So to start, again, we're focusing on the G protein coupled receptor. So we've given an overview and talked about the signaling molecule that binds to the G protein coupled receptor. So that signaling molecule is known as a first messenger. When that first messenger binds to the receptor, it triggers a conformational change and allows for the G protein to bind the intracellular portion of the G protein. And remember that GDP is exchanged for GTP in the G protein. And so that active G protein will now bind to adenyl cyclase. All right. So at this point, that enzyme is responsible for converting ATP to cyclic AMP. And cyclic AMP will relay its signals um, to target proteins. One thing that's important to note is that these second messengers amplify the signals inside the cell. They're produced in large quantities when the receptor is activated, and the last molecule in the signaling chain activates the final response. And so we're going to go over this again in the next slide. So we talked about the first messenger, that's the ligand that binds the receptor and causes the conformational change. But we're more interested in focusing on these second messengers. So what happens in that signal transduction? So what happens in that particular portion of the pathway? So again, cyclic AMP is a second messenger. And we'll go over the pathway more slowly for this. So again, your ligand will bind the G protein coupled receptor. The G protein gets activated. Okay, so the G protein will get activated. And then that active G protein will bind to adenyl cyclase. In this figure, this is the inactive form of the G protein and the inactive form for the G protein coupled receptor because nothing's bound. All right. So again, this is the active form of the G protein. So G, the ligand is binding to the G protein coupled receptor. You're activating this G protein when it separates. Part of the G protein will bind the G protein coupled receptor. Again, the GDP is switched to GTP. The active GTP subunit will bind to adenyl cyclase. Adenyl cyclase catalyzes the formation of cyclic AMP. Okay, that's the second messenger. And this will in turn activate protein kinase A in this case. So remember, your kinases play a role in phosphorylating proteins. So they're going to activate target proteins. And these phosphorylated proteins will eventually go on to alter some cellular process or cause a response in the cell. Now, some of your G proteins will go on to activate other processes in the cell. So some of these G proteins will use phospholipases as second or phospholipids as second messengers. So in this case, you have a ligand binding to your G protein coupled receptor. It's going to activate the G protein, but in this case, it's going to bind phospholipase C. All right. So phospholipase C is an enzyme that's going to cleave phosphate and acetyl phosphate. So in this case, this is abbreviated PIP2, and this molecule is made up of IP3 and diacylglycerol. Okay, so phospholipase is going to cleave this molecule into IP3. IP3 is a second messenger, all right? This will in turn bind to the endoplasmic reticulum and cause an efflux of the calcium ions, all right? And then this will eventually alter some cellular activity. Now, DAG, however, will go on, or diacylglycerol is a second messenger, and it will go on to activate protein kinase C. All right. So remember, our kinases are involved in phosphorylating things. All right. So this phosphorylated protein will eventually activate some cellular activity. So altogether, we have IP3, DAG, all of these are second messengers, along with calcium. That is also a second messenger. Okay, so what are these calcium ions used for? So calcium ions are used for neural signaling. They typically bind to proteins such as calmodulin, and these will help regulate metabolism, muscle contraction, memory, inflammation, and a bunch of other processes that are listed here. So we've gone over some of the second messengers and how they are responsible for amplifying the signal or amplifying cell signaling processes. And another way that you can increase the efficiency of this is for some of the proteins to go on and bind to scaffolding proteins. So your scaffolding proteins are involved in regulating the activity of protein kinases that are used to basically activate multiple signal transduction pathways. And so your scaffolding proteins will pretty much organize groups of signaling molecules. That's their whole function, is to organize groups of signaling molecules, and then you're going to elicit a certain response, okay? So these overall will increase the efficiency of the response. Another way for signaling to occur is via integrins. So integrins are transmembrane proteins that transduce signals in two directions. So you're going to activate signaling molecules when they bind to integrins, and then integrins can in turn respond to information received from the inside of the cell. Okay, and that's going to affect how other molecules bind to the actual integrin. And then finally, do not forget about signal transduction that occurs with the intracellular receptors. Okay, so the ligands again have to be hydrophobic so that they can cross the plasma membrane. And these ligands will bind to receptor complexes on specific regions of the DNA, and they're going to either activate or repress genes, okay? And so these genes will then be transcribed to messenger RNA, and then they are going to be exported into the cytoplasm, and proteins will be made in the cytoplasm. Okay, so overall, we have traced the sequence of events in signal transduction as it relates to these G protein coupled receptors and how these G proteins go on to activate enzymes that eventually activate second messengers in the cell. One of the enzymes that we've gone over is adenyl cyclase. That will be responsible for converting the ATP to cyclic AMP. Cyclic AMP is the second messenger that will go on to activate other proteins to amplify some cell activity. Additionally, we said the G protein can also activate enzymes such as phospholipase, which is responsible for cleaving PIP2 into IP3 and DAG. These are second messengers. IP3 will bind to endoplasmic reticulum and cause an efflux in calcium ions. Calcium ions are also second messengers. Whereas DAG, or diacylglycerol, will go on to um, activate protein kinases, which will then in turn phosphorylate other proteins, and these will lead to the activation of other cellular active processes. Now, to complement this, these processes that we've talked about in terms of the second messengers and amplifying the signal, there are other ways to increase the efficiency of the signal aside from second messengers. We also talked about scaffolding proteins. These are going to organize other proteins onto them, and then that way you can also cause a cascade of signaling events. And that pretty much sums up this learning objective. And one final note is don't forget about the integrins. Integrins can transduce signals in two directions, all right? Integrins can transmit or transduce signals in two directions.